Hello and welcome. This is the 25th Annual Wilderness Issues Lecture Series. I'm Lori Young, the Interim Director of the Wilderness Institute at the College of Forestry and Conservation here at the University of Montana. I'm pleased to present our speaker for this evening, Scott Mills. Scott works here at the University of Montana. He's an Associate Professor of Wildlife Population Ecology in the College of Forestry and Conservation. He's been with the Wildlife Biology program since 1995, and he has a PhD in biology from the University of California, Santa Cruz, where he studied with leading conservation biologist Michael Soule. Scott has authored or co-authored more than 60 professional publications related to wildlife population dynamics and presented more than 50 invited talks at universities and professional meetings both nationally and internationally. His research uses DNA analysis and population ecology concepts along with field research to look at human-caused changes and their impact on wildlife populations. Scott is interested, among other things, in the mechanisms and consequences of wildlife population fragmentation, which makes him an ideal speaker for this series, where we're looking at how to conserve wildlife, among other things, across a landscape. Scott's field research examines, among other things, the responses of birds and mammals to highways, fires, and forest thinnings, and he looks at how to better design corridors to connect populations. Scott's talk tonight provides an important ecological perspective on connectivity and wildlife movement in fragmented landscapes. So please welcome Scott Mills.
Well, thanks, Lori, for that introduction, and thanks to all of you all for coming out tonight on such a beautiful Missoula spring afternoon. It's a little tough to be in here, I know. Studying animal movement is every bit as exciting a scientific endeavor as studying the planets or the stars or the bacteria that chew on sulfur in deep-sea trenches to make carbohydrates without sunlight. On the one hand, a profound human desire for order and a balance of nature have led us, humans in general and biologists in particular, to underestimate the role of dynamic change and how movement could be a critical player in sustaining populations that blink on and off across the landscape. On the other hand, we have a deep theoretical understanding for appreciating the effects of movement among wildlife populations, stretching back to fundamental work by population geneticists and ecologists as far back as the 1930s and 1940s. But the limiting factor has always been lack of data from the field. Animals on the move become ghost-like. They cease to be captured in live traps. They disappear from observation. They fade away from the pinging headphones of traditional radiotelemetry studies. And so for the last few decades, as our theory has continued to build a compelling case for the importance of movement to the dynamics and persistence of populations, wildlife biologists have collectively shaken our heads, shuffled our feet, and despaired that we just don't know very much about how and why animals move. But that's changing rapidly. Recent advances in techniques have revolutionized our ability to study animal movement. And in virtually every new species studied, using these new techniques, we become surprised at how much movement is occurring. And as we understand more about animal movement in a human-dominated landscape, we learn bits of insight that will help make management and conservation decisions a little better. So with that, I'm going to do the Statue of Liberty thing. I guess I didn't. Do it again, Lori? Yeah. Okay. So the creatures that make up my title are, of course, humans, because everything I do has to do with wildlife interactions in a human-dominated and a human-perturbed landscape. It also includes 3 species that I and my graduate students have been spending a lot of time researching over the last few years. I'll also talk about a couple of other case studies that have been done by other researchers. And just because there may be some of you in the audience that thrive on having a little bit of controversy to chew on during the whole talk, I'll give you just a little bit here at the beginning, and that is to say that I'm going to make the point that corridors are a poor substitute for thinking about connectivity, and I'm going to argue that connectivity is not always good for wildlife in a fragmented landscape. So what do I mean by connectivity? I intentionally use this word. It's vague, it's broad, but it encompasses all the pieces or the bits of information that we want to know about when we think about movement across a landscape. Connectivity includes both movement and reproduction. And of course, there's some obvious benefits to connectivity. Connectivity provides access to food and mates. It allows animals to recolonize patches that have become extinct. It minimizes the chance of local extinction for two reasons, two obvious reasons. One is that as a local population becomes small, immigrants into that population take that population away from the boundary of zero, and so therefore decrease the chance that that population will bounce to extinction due to environmental variation or other sorts of random effects. Also, new individuals coming into a population bring with them new genetic material, so that if a small isolated population is suffering from inbreeding depression due to the loss of heterozygosity, that can be broken by the introduction of new genetic material. But we want to not be glib when we think about connectivity, so we want to also recognize that there are potential disadvantages to connectivity in a fragmented landscape. Connectivity can facilitate the movement of exotic species, or the movement of what I call human-facilitated species, native species that reach inordinately high numbers in a human-dominated landscape. And I'll give an example of one of those. And of course, diseases are similar to that. Connectivity in a fragmented landscape can lead to movement into a patch type or movement into an area that reduces survival or reduces reproduction. And a potentially more subtle downside of connectivity is that if populations have become locally adapted to a particular area, locally adapted to particularly cold winters or high selenium content in the soil or a certain moisture regime, movement into those populations can reduce the ability of the local population to adapt. It can essentially break the local adaptation. So how do wildlife biologists measure connectivity? Well, the sort of classic way, the way that puts wildlife biologists on the cover of National Geographic is with radiotelemetry. This is a picture of Kara McCoy, a wildlife biology graduate student, putting a GPS collar on a grizzly bear. And GPS technology and radiotelemetry technology is indeed changing a lot and is indeed increasing our ability to study movement in animal populations, but there's also been some other really radical breakthroughs that are equally responsible for our increased understanding of animal movement. And that would include new approaches to analyzing mark recapture data, and I'll spare you the math, but you can just believe me that there's been some terrific advances in mark recapture analysis, and also the use of genetic tools. I won't be talking in my talk tonight about another sort of research love of mine, which is the use of non-invasive genetic sampling to study wildlife populations using little bits of hair or little bits of fecal droppings or any feathers, any bits of tissue that you can obtain non-invasively in the field can be analyzed, and you can obtain information on relative population size, species that are in an area, and even potentially understand something about isolation and connectivity. I won't be talking about those uses of genetic tools, but I will be talking about the use of other tissue samples that can help us understand connectivity. Okay, so the animals that I'll be talking about today, as I mentioned, include Canada lynx in western North America, snowshoe hares in Montana, deer mice in Oregon. These are species that we've studied, but I'll also throw in some tidbits on Columbia spotted frogs and wolves. So the first story I want to talk about was one that I did with my student, Mike Schwartz, who's finished his PhD a couple of years ago, and he's now a research scientist at the Rocky Mountain Research Station here in Missoula. And the story with Canada lynx really centers on the fact that Canada lynx are, as we all know in Missoula, I gave a talk in UC Santa Barbara a few weeks ago, and I had to explain a lot more about lynx to them, but they're in Santa Barbara. So you all know about lynx, and you know that they're threatened. And you may or may not know that lynx are also sort of the poster child for ecological textbooks for a couple of reasons. One is they have these remarkable population cycles. These cycles sort of occur regularly with 10 years between the peaks and the troughs of the cycles. They're fairly large amplitude between the low of the cycle and the high of the cycle. And they're also synchronous across space. That's to say that a peak in one area tends to be followed very closely by a peak in other areas. So lynx cycles are more or less synchronous across most of North America. And the reason or the basis for this synchrony across large spatial scale has been the source of a lot of discussion. One camp believes that the synchrony in lynx cycles is driven by external factors. This might be sunspots, or it might be common density-dependent structures among the populations and a correlation with environmental events like weather. But in either case, if the synchrony in cycles is driven by external factors, it means that conservation of lynx in the U.S., where of course lynx are listed, they're obviously not they're not listed in Canada, they're not listed in Alaska, but the conservation of lynx in the continental United States is going to need to focus mainly on local populations. But the alternative view is that the synchrony in cycles across space is driven by movement from the northern core. So for lynx, when I say the northern core, here in Montana we like to think of ourselves as being northern, but from a lynx or a snowshoe hare point of view, we're actually southern here in Montana. The northern core is British Columbia, Alberta, Alaska. And so the other view is that movement from the northern core following a lynx peak sends out dispersers that then synchronize other populations sort of in this traveling wave fashion. And if that's true, if movement is the driver of synchrony in lynx population cycles, then that indicates that here in the continental United States we need to focus more on connectivity, or at least as much on connectivity as we do on the local populations. And I think it's fair to say that the dominant paradigm has been that there is very little movement into the United States from the northern core. So this quote from Kohler and Aubrey says, "The conservation of lynx populations is of greatest concern in the western mountains of the conterminous United States at the southern periphery of the species range. Recruitment is low in this region and many lynx populations are geographically isolated." So because this is an important topic both from the point of view of basic ecology and also the conservation of lynx, we waded right into it with a study using DNA from captured or killed lynx. Now I want to be clear that we didn't capture or kill any of the lynx used in this study. Lynx, of course, are trapped. There's a quite healthy trapping season on lynx in Canada and in Alaska, so a lot of our samples were from legally trapper-killed samples, and we also got a lot of samples from biologists who handled lynx in the normal course of their duties. But using those tissue samples, we were able to ask whether lynx at the periphery of their range, for example in the western United States, were in fact isolated from other populations. And related to that, we could ask whether lynx at the periphery of their range tend to originate from nearby populations. That is, do lynx tend to just trickle between populations, or is there a fair amount of long distance movement? Sounds like thunder every now and then. Keep us on our toes. So this is a map of the range, at least the historic range, as best as it can be recreated in blue, the historic range of lynx. And the yellow dots are populations where we obtained about 600 lynx tissue samples. The purple dot here, or pink dot, is a population in Wyoming that we in particular wanted to know whether these animals in this population were more likely to have arisen from nearby populations, such as Sealy Lake, Montana, or from populations further up in the core. So, a lot of people don't know how genetic tools work to understand connectivity. So, this is my best rendition of a lynx. No, actually not. I'm bad, but I'm not quite that bad. This is a slide that was done by a former master student of mine, Reese at Yale, and so I just thought it would be sort of fun, because I don't really know what they are. But the idea here in this cartoon is that, let's see if I can do this without... Yeah, the idea is that you could think in the most simple way of two genotypes, the orange genotype and the red genotype. And so it's pretty easy to see that populations that don't have very much movement among them would tend over time to have characteristic genotypes within the population, or to use the parlance of population genetics, populations will tend to be differentiated. Allele frequencies will tend to be differentiated in populations that are isolated, that do not have much connectivity. And by contrast, a population with quite a bit of movement among populations is going to be expected to have lower differentiation, more homogeneity in genotypes between the two populations. So, of course, the whole process of using DNA to infer connectivity is more complicated than this, but this is the basic idea behind it. So we used 9 microsatellite markers, which are sort of the current marker of choice, to analyze genetic variation and look at the extent of differentiation among these 17 lynx populations. And the bottom line was that we found that gene flow was high among populations, that it was likely to have been high historically, and you can do that with genetic tools. You can look back into the past to some extent using genetic tools, because the genetic architecture of a population today is in large part a consequence of matings that have occurred in the past. And so you can look back into the past using genetic tools, and you can also use a different suite of genetic tools to ask how much movement is going on currently. And using both of these kinds of approaches, we found high gene flow among populations. Furthermore, we found that movement is often long distance. For example, those Wyoming individuals, that group of individuals from Wyoming was more likely to have arisen from parents up in the core of the lynx range than it was to have arisen from, for example, populations in Montana. So movement is happening, it's happening quite a bit, and it's happening across long distance in many cases. So we believe that lynx populations are in fact not isolated in the continental United States. And our argument was supported by some work that Kevin McKelvey did. Kevin is here also at the Rocky Mountain Research Station in Missoula. And he studied the time series that are readily available from trapping data. And the blue line, or actually the red line, is just an index of numbers, minimum number of, or proportion of maximum lynx trap. You can just think of this as an index of lynx numbers. The red bars being lynx numbers in an index of lynx numbers in Montana over time, trapped based on numbers of trapped lynx. And the blue data are the numbers of lynx in the core, in the British Columbia, Alberta core, shifted by 2 years, shifted forward by 2 years. And in fact, what Kevin found was that the further you go away from the British Columbia, Alberta core, the more you have to shift the core index data to have it overlie the populations that are further and further away. So what that indicates to us is that lynx build up in this northern core, then they disperse out following the peak, and so you get this traveling wave that goes across the country, goes across the continent really, and so you get a lag time between peaks, with the lag increasing as you go further and further from the core. So this indicates to us that lynx populations are highly connected, and obviously that's going to be important for recolonization of patches that go extinct in the continental United States, for example. It would also be important for rescue effect for increasing numbers of very small populations that are in the western United States, taking them away from that boundary where extinction is likely. and in fact it may be a driver of synchrony across space. So the implication for this that we thought was most striking was that endangered species management has done a fairly good job of focusing in on endangered populations. We have things like critical habitat that focuses in on the populations themselves. But we haven't been so good about thinking about what about protecting areas where the animal isn't even found, maybe where there's no presence of the animal, but it's a critical area for the animal to use as it travels across the landscape. And so we made the argument that managing movement routes may be as important for lynx conservation as managing the extant populations themselves. That's not to say that it's unimportant to manage existing lynx populations, but actually managing the movement paths for lynx may be as important as managing the populations themselves. Okay. From lynx to frogs. Frogs are sort of this classic species that we all think about as just staying in one place. I mean, what does a frog do? They sort of pile on top of each other, and then they swim around in a pond, and they hop up on the edge, and they sort of they're just frogs, so they just stay around in one place, it would seem. And so the story I want to tell here is work done by Chris Funk, who recently defended his PhD in biology and kindly loaned me some of these slides, like I said, almost every time we go out and look at animals to study movement, we find more movement than we thought possible. And you might guess that that's the story I'm going to tell you here. So Chris used heroic mark recapture work, marked over 10,000 frogs, and he also used DNA analysis to study movement. And I'm going to just talk about some of the mark recapture results. He did this in two areas of very western Montana, Keeler Creek and Martin Creek. And the punchline is that he found that these frogs move. This is just a histogram that shows this is zero movement, and going out from zero is movement in one direction or the other. It's just frequency of animals that moved different distances. And the punchline of this is that 14% of juvenile frogs moved more than a kilometer, and 2% of adults moved more than a kilometer. But that's not nearly as striking as this. So Chris found two frogs that moved from this area to this area, which was 4,240 meters, 770 meter elevation gain. And if you're not used to the metric system, that's a movement of 2.6 miles for these two frogs. Little bitty frogs, lots of hops. 2,526 vertical feet, a half a mile of elevation gain. So you might think, well, that's okay, but that's just two frogs. But from over here, he had four frogs that moved essentially the same distance and essentially the same elevation gain. And that's not all. Two more frogs that moved not quite as far, but still substantial, almost 2,000 meters and 700 meters elevation gain. So these frogs are moving. Not only they move to where they move to a new place, and then they stay there. And so it seems pretty clear that movement becomes very important for frog population dynamics. And this is also counterintuitive because we tend to think about, and the global amphibian decline community has focused on frogs, for example, or amphibians in wilderness areas. And when they've seen declines in wilderness areas, people are always scratching their heads and saying, well, this must be some big global factor. This must be acid rain. It must be UVB that's causing these kinds of declines, because these are, after all, wilderness areas. So how could frogs up in these wilderness areas be affected by anything? Because they're in a wilderness area. But when you realize that movement is actually pretty substantial for frogs, then it doesn't take much to reason that if something happens to the low elevation population, which, as we all know, are much more susceptible to perturbations, or something happens to, again, the movement pathway that frogs might take as they move from low to high elevation, then you could see effects on the high elevation populations that are completely non-intuitive based on just studying the high elevation population alone. And then for a sort of a completely different example, but to kind of round out this discussion of some of the benefits of connectivity, the last story I want to tell you about was done by some folks in Scandinavia. The story with Scandinavian wolves is very similar to the story of wolves in the United States. They were very common, and then they were extirpated, and then they have slowly been coming back with the cessation of hunting. The Scandinavian wolves are slowly coming back in many areas of Scandinavia. So there was this one particular place in Sweden where 2 wolves arrived, and then no more wolves arrived for a bunch of years. And the population just limped along. And then a new male arrived. And this graphic is a little bit lame. It was scanned in and didn't show up so well. But the main thing you need to pay attention to is the dark line, which is just the total number of wolves in this population. The bars are number of packs, but you can really just focus on the number of wolves in the population. So here they were founded in the early 1980s with these 2 wolves, limped along less than 10 individuals for more than a decade, which, as we all know in Montana, is not very rapid wolf population growth, since we had here in Montana a tripling of population size every year for 10 years once wolves were reintroduced into Montana-Idaho population. But these wolves just stayed down, stayed low. Then the single immigrant arrived, and these guys did genetic studies to show that that individual immediately bred. Its genes were spread throughout the population. That is to say, its genes rapidly spread through the population, and the population very rapidly increased. At least consistent with a hypothesis that this small wolf pack may have been suffering from inbreeding depression, and then with the arrival of this single male immigrant, that the inbreeding depression may have been broken and leading to a rapid population increase. So, using these 3 different stories, I hope to have convinced you that certainly there is major benefits to connectivity for wildlife in a fragmented landscape. Obviously, access to food and mates, recolonization of extinct patches, and decreasing the chance of extinction via numerical rescue and the input of new genetic material that could break inbreeding depression. But as I promised, we need to also look at the other side and realize that connectivity can have distinct disadvantages as well. So next I'd like to talk about a couple of cases that point to some of these disadvantages. I've been studying snowshoe hares in western Montana for about 6 or so years. We've recently expanded. We're now working in Yellowstone. We'll be working in Glacier starting next year. We're working out in the Flathead Forest and in Okanagan, Washington. But the part I want to talk about today is some work that was done with Paul Griffin, who defended his PhD last year. And maybe some of you are taking ecology from him this semester. He's teaching the general ecology this semester. This is our study area in the Seeley Lake area, looking, of course, towards the Bob Marshall. And as we know, the Seeley Lake area, like western Montana, doesn't have a lot of human dwelling pressure, but there's certainly effects of logging on the landscape. And this is just another shot of the study area. And the story with snowshoe hares is that, like lynx, they also, in the northern area in Alaska and Canada, undergo remarkable population cycles, also 10-year cycles. There's almost certainly an interaction between lynx and snowshoe hare because hares are the main prey of lynx. But for a long time, people have thought that maybe patchiness or fragmentation in the southern part of the range, again, that's the U.S., that the southern part of the range that patchiness in the southern part of the range may affect hare population dynamics. And the thought is that possibly, but it's never been tested, is that possibly snowshoe hares start to build up abundance in these closed canopy dense areas that hares tend to prefer, and then hares disperse out into the open areas, and as they disperse out, there's a suite of generalized predators waiting for them. The predators gobble up the hares, and that essentially chops off the top part of the cycle. And so we set out to test that, and by studying hares in closed canopy, what we call closed canopy young forests that might occur, for example, after pre-commercial thinning, closed canopy older forest with multi-layer canopy that might be expected in a mature or old growth forest, versus open areas that might be open soon after pre-commercial thinning or open after selective harvesting or quite a few years after pre-commercial thinning. And One thing I have to say about snowshoe hares is they get killed and eaten by everything out there. Snowshoe hares don't die of old age. They get killed, of course, by lynx, but also by coyotes, goshawks, great horned owls. Anything that eats meat out in the landscape will eat snowshoe hares. And actually, other things will too. Like it turns out that, little known fact, red squirrels are one of the greatest predators on snowshoe hare babies. I always like to use that when I teach general ecology because the good old classic food chain has a red squirrel as this cute little herbivore, but it turns out that they just clobber baby snowshoe hare. So think about that next time you're eye to eye with the red squirrel. But one of the most embarrassing things that can happen to you as a field biologist is to be following perhaps a radio-collared snowshoe hare and to come through the underbrush and come face to face with this. Because this hare thinks that he is perfectly well camouflaged. You see, the snowshoe hare coat color change comes from the pineal gland deep within the brain of the snowshoe hare, and that's driven by day length. So day length tells the pineal gland when to change the coat color from brown to white and from white to brown again. But as we know here in Montana, day length does not necessarily correspond with a consistent snowpack. And so you get these cases where snowshoe hares have this mismatch, and they think they're perfectly well hidden. They're just sitting there, and you just kind of turn away and hum a song and something because you're so embarrassed to see them in that way. But they get clobbered. They get clobbered. Their highest mortality rates are in the spring and in the fall. And the highest mortality rates in the spring and in the fall, which we think is at least partly driven by this mismatch, is not constant across these different forest stand structure types or these different habitat types. In particular, what we've got here are the different patch types, the dense mature, the dense young, and then the open young and open mature. And this is population growth rate, which all you really need to know about it is that dotted line represents a stationary population, neither increasing or decreasing. Above the line is an increasing population, below the line is a decreasing population, and the black bars are confidence intervals that take into account the uncertainty in estimating birth and death rates that you have to take into account to estimate population growth rate. And we can see that the dense stands, the two dense stands, are both overlapping a stationary population, but the open stands are representative of declining populations. That is to say that hares that remained in the open populations would be expected to decline over time, have negative population growth due to decreased survival and or decreased reproduction rates. Oh, this one didn't come out so well on the PowerPoint conversion. It's okay. This really just shows, all this shows is movement of hares. So hares don't stay in those particular stand structure types. They move around a lot. So this is the relative proportion of snowshoe hares that use the dense mature exclusively. This is the relative proportion of snowshoe hares that use the dense mature not at all or very little. And you can see that most hares use several different patch types. So hares are moving around in this landscape that includes good areas for them, areas where survival is high, reproduction is high, where population growth rate would expect to be positive, and includes areas where population growth would be expected to be negative. And so for snowshoe hares, this local movement, this movement on relatively small scale or longer movement, and we have a radio track snowshoe hares moving 3 kilometers or more. So movement of snowshoe hares in this case across this patchy landscape that has areas that are more or less likely for snowshoe hares to be killed in, this movement across this fragmented landscape has the potential to dampen cycles as movement sends individuals to their deaths in these more open areas. I mean, it can be stated dramatically, but that really is what's happening. Okay, and then the last story that I want to talk about has to do with deer mice in Oregon. I studied several fragments in Oregon surrounded by clear cuts several years ago, and then actually about 15 or 20 years ago, and then I had a PhD student, David Tallman, who went back to the same areas and looked specifically at the question of deer mice and Trillium. So these are just some examples of a couple of the fragments that we studied in Oregon. And Trillium, you've got to know, is just the coolest plant. I mean, of course, we have it here. It'll be blooming very soon in places like Woods Gulch, and you should really go out and bond with Trillium. These are amazing plants. They're, the species in Oregon that we studied is exactly the same species as here. They live to be 70 plus years old. So I always tell students that they could be like your grandparents' age or more. These Trillium could be. And you can tell that because people like Eric Jules, who's currently a professor at Humboldt State University, have figured out that you can very carefully dig up Trillium, or at least move the dirt away from Trillium, go down to the roots, look for constrictions on the rhizomes that are annual constrictions, count those constrictions, age Trillium quite accurately, cover them back up with dirt, and the Trillium's fine. So you can age them without hurting them. And if you do that, you find that, and also what Eric has done is coupled current density estimates of Trillium that he's done with historic estimates of Trillium that have been done in the past. And from that, he's found that densities now on these clear cuts in southwest Oregon are much lower than densities were before logging. And on the fragments themselves, densities are still holding their own. That is to say, densities on the fragments are about the same as they were before logging. But the really interesting thing is that the Trillium on the fragments are old Trillium. There are almost no Trillium on these forest fragments, patches of forest surrounded by clear cuts. There is almost no Trillium younger than the age of the surrounding clear cuts. And work that we've done and work that we've done collaboratively with Eric implicates deer mice. You're supposed to all hiss when I say deer mice. Implicates deer mice as a driver of the limited recruitment of Trillium. Deer mice are voracious seed predators, and they respond very well to opening up areas in, for example, using logging. So, for example, we studied this particular graph shows 3 forest fragments that we studied over 2 years and 2 nearby control areas in and around Oregon Caves National Monument. And you can see that consistently the forest fragments have higher densities of deer mice than the control areas do. And I haven't shown you here, but it also appears that deer mice are more abundant in clear cuts than they are in fragments. So deer mice numbers are quite strongly control, with a lot more in the fragments, and then even higher density in the clear cuts surrounding the fragments. Okay, so I've got to bring this, it's a cool story, but you're wondering, bring this back to connectivity. So here it is. This is using mark recapture analysis. This is using these very cool new advances in it's called multi-state mark recapture analysis, where you can make a smiley face. Well, no. You can make a diagram that actually shows the proportion of individuals leaving each geographic state and moving to another geographic state. So it's possible to actually quantify using patterns of marking and using patterns of recaptures to quantify the probability of an animal in one area moving to another area. So what you can see is there's quite a high proportion of mice moving from the interior out to the edge and from the edge out into the clear cut. There's a reciprocal movement from the clear cut back to the edge. Fairly high movement from the interior. This is the interior of fragments and the edge of fragments, by the way. And also quite high proportion of animals moving from the interior out to the clear cut. Lower proportion moving from the clear cut back to the interior, but since numbers are actually high in the clear cut, there's actually still a fair number moving back into the interior. So numbers are high. These animals are subsidized by logging. They're doing very well in this fragmented landscape, but they're not just static. They're not staying in one place. They're moving around, and they're taking their little teeth with them. So forest fragmentation has changed the landscape, and the resulting clear cuts does seem to be serving as a demographic source for these voracious seed predators. And so in this case, connectivity is just part of the player, but connectivity has a role in causing these cascading effects where a human-facilitated species, deer mice, a native species, perfectly native species, but a species that's subsidized by humans that does very well in a human-modified landscape, and it then has cascading effects on other species, like Trillium. Okay, so I've sort of painted for you this tension or this dynamic between, on the one hand, the benefits of connectivity, and on the other hand, the downsides of connectivity. And so it leads to the natural question, well, how much connectivity is enough, or what level of connectivity is appropriate in a landscape? And if we're restoring connectivity to a disturbed landscape, what level of connectivity should we be striving for? And it's certainly a very fair question. It's a challenging question, although a lot of people have thought about it. One thing you could do, the sort of common sense thing to do, would be to estimate natural rates of connectivity, try to estimate background levels of connectivity in an unfragmented area, and then compare that to a fragmented area and say, well, what we need to do is get connectivity in the fragmented area up to the same level as this control area that wasn't fragmented. And that can be done sometimes. It was done by some folks up in Canada who compared genetic structure for grizzlies with and without highways and concluded that the highway corridor does decrease movement. By the way, I haven't talked about it, but we've worked on small mammals in Montana and looked at effects of highways, and certainly highways can play a role in affecting connectivity of small mammal populations. So I want to be sure to not make it seem like logging is the only force that perturbs or fragments wildlife populations. Certainly roads and subdivisions can do that quite well, and Walmarts can do that a lot as well. Had to slip that one in. The problem with using this approach to estimate connectivity is that, as you know, there's not very many landscapes that are big enough to serve as a control for understanding connectivity of wide-ranging species, birds or, for example, large carnivores. And so this just won't work in a lot of cases. I mean, you're not going to be able to obtain movement on wolverines in an unfragmented area. There's just nowhere that's big enough. And so sometimes we fall back on rules of thumb, and the most used rule of thumb out there is one that was based purely on genetic considerations. And that is that 1 to 10 individuals moving among populations per generation, not per year, but per generation. So you can figure 3 to 6 years for most carnivores, for example, for a generation time. That movement of 1 to 10 individuals per generation is enough to reduce the chance that inbreeding depression will occur while still allowing populations to locally adapt to the conditions where they're found. Based purely on genetic reasons, plenty of reasons why you'd want more than 10 migrants per generation or less than 1 migrants per generation once you start weighing out the pros and cons of connectivity, but that's a rule of thumb that's often used. So that brings us to the question of how to achieve connectivity. And the first thing I'll say on that is to avoid platitudes. I think that corridors, the concept of corridors can be useful for initiating a conversation about the need for large-scale regional connectivity. But I think what happens too often is that people say, well, what we need is just more corridors. And then the brain sort of turns off. And a linear strip of a particular kind of habitat could easily be a death trap for certain organisms. It could easily be the kind of habitat that exotic species or human-facilitated species like deer mice would use, or brown-headed cowbirds would use, or other nest parasites or nest predators. And so linear strips that we often think about for corridors that look kind of nice on a GIS map where you have patch A, patch B, nice green corridor in between, that may be a good way to connect landscapes, but I think it's better to focus on connectivity, to ask what exactly do you want, what are the species that you want to benefit from connectivity, and then how might you manage the landscape. This is often referred to as managing the matrix, managing the surrounding stuff that is between patches, and as a more efficient and thoughtful way to manage for connectivity. Okay, so concluding from all this, what do we conclude from all this? First of all, like I mentioned, every time we rigorously study connectivity, we find that animals really are moving more than we thought was possible, more than we ever imagined. So that's one take-home message. Another is that clearly, both theoretically and empirically, it's clear that movement can have big consequences for native species. Movement can be positive, movement can be negative. a corridor could be positive, a corridor could be the worst waste of money that you've ever imagined. I mean, both of those are possible scenarios. And changes in the landscape is going to decrease connectivity for some species and increase connectivity for others. So again, I think that you can't really think of I get really queasy when I hear people talk about corridors to increase ecosystem health or something like that. I mean, you can't just have sort of a one-size-fits-all corridor or one-size-fits-all recipe for connectivity, because how you manage a landscape is going to affect connectivity for different species differently. And so there's, once again, my little rant about corridors. And so with that, I want to leave plenty of time for questions. Since my 3-year-old, Nicholas, this is for you, came to the talk today, I wanted to have Dr. Seuss, Dr. Seuss, of course, anticipates every scientific debate, and this is Dr. Seuss's view of connectivity of wildlife populations. So I wanted to give Dr. Seuss the last word here. So thanks. Yeah.
What are your views on the Yellowstone to Yukon?
Well, I think it's visionary, and I think it's. I think the idea in general of thinking about connectivity at really large scales is the way we have to go. We have to be thinking about that. I mean, everything from current changes to global warming tells us that it's not a static world. Populations probably never stayed in the same place, never persisted in single populations, and so we've got to be thinking about a dynamic landscape and thinking about connectivity. So, so I definitely think that the whole sort of wildlands sort of philosophy is an important one to have on the table. And of course, it was co-created by my PhD advisor, Michael Soule. But I've had a lot of debates with him about that. I mean, I think that in some ways, the Wildlands Project has led to a lot of misuse of this corridor concept. I mean, I debate with him a lot this sort of use of corridors, because I think it kind of gets used too fast, where people aren't really carefully thinking about what they mean by connectivity. But the idea behind it, that we need to be thinking really big scale and really long term for persistence of wide-ranging species, it's almost a no-brainer that biologically it's the way to go.
With the two examples that you had, the snowshoe hares and the deer mice, they're connected to areas where there's been an alteration in the landscape. For one reason or another, it's bad for either the trillium population or it's bad for the snowshoe hare populations. But it doesn't seem like anyone built a corridor from an old-growth forest into a clear cut. Is the problem more connectivity, or is it more in fragmentation or
Well,
the matrix?
Yeah, I mean, it's a good question. And of course, fragmentation, the word itself, and I probably should have been a little more careful about defining that at the beginning, but fragmentation has what I think of as three main parts. Fragmentation involves a loss of habitat, a decrease in the population size. That's problem number one. And problem number two is that fragmentation involves a change in connectivity because what used to be continuous becomes chopped up. And yeah, fragmented like a plate, that gets dropped and the pieces go everywhere, except for it's not quite like that because the sum of the pieces aren't the same as the original plate was. And then the third thing is change in the quality of the habitats that remain or the populations that remain, things like edge effects, that because now the originally continuous habitat is now juxtaposed against a very different structure. So yeah, I mean it's a tough one. I mean, fragmentation does make patches smaller, but it does immediately juxtapose populations exposes them to different stressors than they would have had before the fragmentation occurred. So in a sense, fragmentation by definition does create corridors all the way through. I mean, and certainly in the case of highways, it creates, you know, it's a sort of a corridor that's put through an area. And there has been cases with nest predation of birds where linear corridors that people have envisioned building between patches have been shown to be ecological traps where nest parasitism, for example, is higher in the linear corridors than it is in the continuous areas. So yeah, it's a tough thing to separate out. It's a tough thing to separate out the change in fragmentation from the change in connectivity from the other changes that come with fragmentation. That's a good point.
What's your favorite explanation of link cycles in the core area? Do we have one?
Yeah, well, you know, I think like so many dichotomies, the dichotomies over time was the one camp that said they're strictly driven by predator-prey dynamics, that they're classic Lotka-Volterra predator-prey dynamics, and the other school that says no, they're bottom up. The vegetation gets eaten by snowshoe hares. The vegetation defends itself, puts these chemicals in. The chemicals then start causing hares to die off because they don't get enough to eat, and that then causes links to die off as the hares die off. So, so I think that neither of those are right, and I think that the current view, which is probably the correct view, is a synthesis of those two, where it's largely top-down driven, largely predator-prey interactions, but there's also a component bottom-up where the vegetation isn't a passive player. The vegetation is actively defending itself, and that vegetation changes can cause the vegetation can cause the cycle to turn, but then predator-prey dynamics are going to drive the cycle down or up. So.
And question about the heroic frog study. How this dispersal was quantified? Because the farther they move, I imagine the longer was the distance, the harder was to find those frogs. So
Yeah.
do you think those estimates of long movements were pessimistic?
Yeah, well, so yeah, I mean, you've identified exactly the reason why we haven't made much progress on understanding movement of wildlife populations for the last 150 years until the last couple of decades. Because if all you have is some good old wooden traps that you've built by hand, which is the way people used to catch animals, and maybe some radio collars that transmit short distances, then you really can't. Then movement is always going to be underestimated. You're always going to. You're always going to underestimate movement because long distance movements are going to be away from where you are going to. The radio collars are going to fade away, and so you're going to miss long distance movements, and there's not going to be traps there to catch animals that move long distances. And so that's why for a long time we've just known that, like I said at the beginning, that any estimates of movement that we have are biased low and that we're missing infrequent long distance dispersers. So all of that is by background to say that in some ways Chris was just lucky, and in some ways he chose a really good species where he could mark 10,000 of them. Most species that you can think of, we never would mark 10,000, so we would just miss those movements. So yeah, I don't think that necessarily that's going to show the longest distance movements that frogs are making. But the fact that those distances were detected is pretty striking to that movement is more than we expected.
I just wonder, you showed this histogram of distance of movements.
Yeah, the tail is surely underestimated. Always will be in a pure mark recapture study or even in a telemetry study unless you're able to use GPS collars that are working under all conditions that the animal's moving through. And the batteries don't die and you can afford the data stream. Grant doesn't get cut. Yeah, Jeff.
Scott, on the Colorado lynx reintroduction, was that reintroduction effort informed by McElbee's data, that cyclical correlation?
Oh, people certainly knew about lynx cycles when they did the Colorado lynx reintroduction. If you're pushing me to the controversial question of what happened in the Colorado lynx introduction, I think that perhaps there wasn't as sound an information as would have been good to have on the densities of snowshoe hares. I think that there was perhaps a bit of haste in essentially there was no marker capture that went on to determine snowshoe hare densities. It was based on pellet counts, and I think there was perhaps an underestimate of hares that would be available for lynx. So, yeah, I think that people knew about the cycles, but I think that maybe people just missed on how many hares were there to support them.
Is there evidence that there continues to be a natural dispersal in Colorado where they're reintroducing the lynx?
Well, you know, the Rocky Mountain Research Station folks, John Squires and crew, have been studying lynx for as long as I've been studying hare. In the same areas where I've got hares, where I'm studying hares, they've got radio-collared lynx up there in Seeley Lake, and they have found lynx that have moved down into Colorado and then moved back. So they've certainly shown radio-collared lynx that were capable of moving into Colorado and back, but they haven't had one that went to Colorado and stayed. But again, relatively small numbers, 20 to 25 lynx radio-collared at any one time. That's an awful lot for a carnivore study, but it's not 10,000 frogs. So again, we're probably underestimating the movements. Did you?
Just out of curiosity, how do you mark a frog?
You toe clip. And in case anyone's squeamish about that, it is, I will strongly defend toe clipping. You can't think about how it would be for a human. That's oftentimes what people do, and it conjures up terrible images. But toe clipping, it's clearly the best way to permanently mark most small animals. It's a permanent mark. It does not affect survival, and it has amazingly little effects on even short-term behavior. And it also immediately provides a genetic sample, which everything that I've talked about that can be done with genetic approaches can be done with a tiny bit of tissue from, for example, a toe clip. There are other ways to do it. There's just blanking on their name, the rice grains. Transponders, yeah. Tiny little transponders about the size of a grain of rice that you can inject under the skin and then run a reader over it. But I don't like those. I think those are much harder on the animal. Ear tags people could use, not on a frog, but you could use for small mammals. But I don't like those. You know, there are other approaches for turtles. People use notching of shells. But I think in modern wildlife biology, every time we manage an animal, we're being negligent if we don't at the same time obtain a genetic sample. And it doesn't have to be much of a genetic sample. And so toe clipping for frogs is a good way to immediately obtain a genetic sample that we can use for lots of things, archive for long term, and that we can also use to permanently mark an animal to understand movement. If you cut corners and you don't get a permanent mark, then you just then you've wasted a bunch of money.
Has anybody looked at the genetics of the lynx and whether populations that make it down to the south end go back, or how long those stay for?
Yeah, no, it's a good question, and there comes a level that we just can't address from the genetic data. So we can look at pairwise movements, we can look at pairwise levels of connectivity between those 17 populations, and we find that there's not isolation by distance. In other words, it's not more likely that populations that are further apart are more differentiated than populations closer together. But we can't tell anything about the reciprocal nature of that movement. All you know is that animals came and the animals moved back and forth, but you can't say anything on a timeline of was there movement down and movement up. And another thing people have asked us to do is maybe people associated with the Y2Y project have said, well, we've got these cool maps of potential lynx habitat, these GIS maps. Can we use the genetic data to ask whether lynx are actually moving the places that using the pathways that the GIS maps would predict that they are using? And it's just hard to get resolution. It's hard to get resolution at that level. It may be possible. I mean, you know, genetic changes. The genetic analysis is going so rapidly. What it is essentially becomes a statistical power question, where it becomes hard to distinguish between movements that aren't so different. But at least theoretically, if you could get lots and lots of microsatellite loci, it's like you're increasing your sample size. You could increase your statistical power to detect different kinds of movements. So it may be that in the future, I mean, I think that there's a big future in using genetic tools to understand connectivity, and it may be that those kinds of questions can be addressed, but just not right now. Yeah. Yeah.
Do you think bioregionalism approach to conservation might be effective in connectivity in the future?
So define what you mean by bioregionalism.
Like educating people with more of the local level of human relationships to land and hopes that they take better care of the surrounding environment. And then one of the foundations is permaculture, build a nice land with permaculture. So it's just creating better landscapes.
Okay, well, just so I don't pester you and ask you what permaculture is, I'll say that bioregionalism, that sounds like that's a good idea. I mean, obviously, I think the more you can educate people to your surroundings. I mean, I always like to end my population ecology class with a quote from Aldo Leopold about the educated lady who didn't listen to the geese flying over her house every fall morning, and trading. If education is a process of trading awareness for things of lesser worth, then what good is education? So I mean, definitely, I think don't take that too literally, students. But yeah, I mean, obviously, having people become more aware of their surroundings is step one. Yeah. You guys gonna let this guy ask another question? Okay. I know him. He's in my class, so I'm not picking on him.
Sure, you may be that there's genetic analysis, for example, of lynx movement shows you present state of affair and not something that was 50 years ago or 100, because now it's fragmented with highways and so on.
Yeah.
But those populations can still be similar.
Yeah.
It's a sort of artifact.
Well, so I don't want to get into the details, but there is this new breed of approaches called assignment tests that use that for each individual in a population you can ask, is its genotype more likely to have arisen from this population or this population? So you can ask, given the genotypes of all the individuals in the populations, you can ask which population is that genotype more likely to have arisen from. And so from that you can make a statistical assignment, and if it's misassigned, then after you account for just error in assigning it, a misassigned individual, that is an individual that's captured in a place where it was not likely to have been born, that is, it's found in a population that is not the closest match to its own genotype, then that indicates that either it has dispersed or one of its recent ancestors, its parents or grandparents, have dispersed from a population with a very different genotype into this population. So we can never be sure, but using the assignment test indicated to us that movement was continuing.
I warned Scott that I was going to ask him this question, but I was wondering, Scott, if you could just talk with us a little bit about how some of your research as a biologist, particularly lynx research, was sort of thrust into the political spotlight, and kind of the relationship between politics and biology, and how you kind of continue as a scientist working toward conservation and understanding of population dynamics in the context of bigger political forces that want to use your research in different ways.
How do you not become cynical and bitter? No, Lori did warn me that this was coming, and I'll warn you all that you can leave if I start to rant and go over time. But no, it's in the past now. I'm mellow. So you may or may not remember that a couple of years ago there was a giant brouhaha when we were conducting. I did a study with Kevin McKelvey, a nationwide study of the presence and absence of lynx in 16 states using hair snag pads. And so the idea is you encourage lynx and other animals to rub up against these pads, and they leave hair behind. And then my lab developed a mitochondrial DNA test to be able to tell, even with single hairs, what species left that hair. And we were very careful before we ever initiated this at a nationwide scale, because we knew that there would be 800 or so people that were going to have to collect these samples. There's no way you can do a study across 16 states with 30,000 or so hair pads and not enlist hundreds of helpers. So we made sure that we had blind tested the protocol to tell species apart. We made sure that we had done blind tests internally, externally. We made sure that we sent an extensive protocol to everybody that worked on this project. And somehow, up to 4 people in Washington and Oregon ended up having the idea that it was okay to go to a petting zoo and collect samples from captive lynx, put them on the pads, label the pads as if they were collected in the field, give them a UTM coordinate, give them a slope and elevation, label them as if they were collected from the field, send them to us, and not tell us that these were not samples that were collected as part of the nationwide protocol. We found out about it, and we thought, well, this should be treated as any field assistant mistake. It was a wrong thing to do. If one of my field assistants, say on a telemetry study, had made up data for any reason, I would give them a stern talking to, tell them never to do that kind of thing again, make them sure that they knew the importance of integrity of data. So Kevin McKelvey and I, our tact was to simply, we thought we'll just handle this internally, find out who did this, and make it clear that it's not acceptable. But the Washington Times broke the story in December as a fraud of immense proportions, saying that these guys had intentionally placed lynx hair into a nationwide study in order to shut down logging in Oregon. And in fact, these guys had done these mislabeled samples in national forest where we had not detected any lynx, and in fact where there was no reliable lynx detections for the last 10 years. So it very quickly hit the proverbial fan, and the conservative end of the political spectrum immediately jumped on this as a demonstration that endangered species biologists were in fact a bunch of crooks, and that this demonstrates that they probably made up spotted owl locations, and that endangered species biologists were not to be relied upon. At which point, I guess you'd say, the left side of the political spectrum came to the vehement defense of the people that had mislabeled the samples, said that they were being unfairly maligned by conservatives, and that really all these guys were doing was to conduct a blind test of the lab, and that what they had done was completely appropriate scientific procedure. At which point I remained right smack in the middle, saying that A, what these guys did was completely wrong and was not legitimate by any scientific standard, and B, that I see no reason to think that 4 people screwing up in a nationwide study of 800 people indicated fraud of immense proportions, and that endangered species biologists were a bunch of crooks. So I spent about 6 months answering phone calls from about 50 reporters. 4 congressional committees investigated it. I had like NYPD Blue guys calling me and over the phone, reaching out and bonking me around to try to get me to say things. Well, yeah, there's just so many things. But the bottom line was that in the end, I went to Congress to testify on this, along with Kevin McKelvey and along with a couple of other biologists. And I think that in the end, there was no substance for either of what I consider to be the extreme interpretations of events. There was no substance for the interpretation that endangered species biologists are a bunch of frauds, and that biology can't be counted on to provide informed information to the political decision-making process, nor is there any support for the equally extreme view that what these guys did was just good, honest science, and that they were being unfairly pilloried. And so it was a very brutal process. What Laura's referring to is that you quickly realize that that many times people aren't necessarily looking, and I'll say people from all stripes of the political persuasion are not looking for scientific information, but they're looking for a view that can be condoned by a scientist that supports their own political viewpoint. But I didn't end up emerging from it as a cynic, and I didn't end up emerging bitter, because in the end the process worked about like you'd hope it would. I mean, these guys, it was found out what they did. It did not damage in the long term the study. We're continuing to write up the study, and I think it remains one of the most ambitious and reliable estimate of Canada lynx presence across the entire United States. The studies continued, it retained its integrity, and in the end, there was just no purchase for these extreme political views. But it took an awful lot of time to get to that point. So,
You talk about reliable lynx detection. What's the definition of a reliable lynx detection? I'm going through the lynx EIS right now for the Northern Rockies.
yeah.
And I'm looking at some of the data for some of the national forests. And there was one trapped, a couple seen, but I think it's been like 20 years ago. Where's that?
Yeah, I mean, that's a good question. And it's certainly one we wrestle with when we set up this nationwide study to, at what point does absence of detection, for example, mean absence of the animal? At what point does definite hits of the animal mean that there is a population in the area? I mean, we know they move a lot. We know that there are captive lynx out there that people have, although most of those are Eurasian, and our genetic test distinguishes between Eurasian lynx and Canada lynx. But it's hard to tell Eurasian lynx from a Canada lynx in hand. We also know that some hybridization is beginning to occur, at least in some areas, between bobcats and lynx. So you would certainly not want to say that a single lynx detection, either through a rub pad or through a hunter-trapped animal or through a roadkill, means that there's a viable population in that area. And so I think you need to, I think it is a tough call. And I guess the answer to that question kind of the answer to what is a reliable detection kind of depends on the question that you're asking with respect to detection. I mean, if detection just means was there an animal present there, then that's more straightforward as long as you make sure it's not a Eurasian lynx, you make sure it's not a hybrid, you make sure it's not captive lynx that's gone wild. But it's a little harder to say whether if you ask, does a detection mean a viable population where management should be changed, where logging should be shut down, where those kinds of things. That's a tougher question. So yeah, I mean, I guess to answer your question, it may sound like I'm sort of dancing around it, but I think it legitimately is the sort of thing that you'd want to think long and hard before you gave a specific answer to that question.
Well, you were talking about a one-size-fits-all management type thing in those corridors.
For the movement of lynx?
Yeah.
Well, I think it's sort of one-size-fit-all, just to say in the sense that, I mean, it's not one-size-fit-all in terms of exactly how that would be implemented, but just one-size-fit-all in terms of I'm quite confident that if you put a Berlin Wall across the Canada-U.S. border and you kept lynx from coming in, I would put money that in 20 to 50 years that I would guess that there'd be very few lynx left in the United States, just because I think that a lot of the lynx that we have in the United States are pulsing down from Canada. Somebody could do an experiment for that and test it.
What's going on in Canada as far as management that's different from the United States?
Well,
We're getting on the southern edge of that.
that's part of it. Yeah, sure, that's part of it. I mean, it may be that if you could look back in a crystal ball and look at what were lynx dynamics 200 years ago, it may be somewhat similar to what it is now. So part of it is just the core versus the periphery. Part of it may be, well, the higher snowshoe hare densities, and you could ask why are higher snowshoe hare densities, and that probably comes back to the patchiness of the landscape as I mentioned for the hare story. So, I mean, clearly at snowshoe hare highs and even snowshoe hare lows, our highs are nowhere, the highest that we ever get in our snowshoe hare studies in Sealy Lake is nowhere near the highs for Canada for snowshoe hares. And our lows are low. I mean, we're getting, in a lot of our sites, we're getting lots of zeros. So I think just there's not as many snowshoe hares in this part of the world. And that affects lynx.
Are there any known disadvantages of connectivity for visitors?
Well, I don't know. I mean, yeah, I guess I don't know. I don't know the answer to that. Karen was going to come. Karen McCoy, did she come? I guess she didn't come. I don't see her. I mean, certainly grizzly bears occasionally get killed on highways. Certainly they occasionally get killed for other reasons as they move long distances. So, I mean, certainly any long distance movement for any carnivore could expose them to a whole suite of reasons why they might get in trouble and get moved or get shot. And I think that's true for any large carnivore. Yeah, so I would think so. I would think there could be, but yeah, I don't know.
Any final questions, Ms. Scott? Okay, thanks very much.
Okay.